Welcome to myotherprofessor.com. I'm Zaina, and today we are going to discuss cell theory. So, brief outline of the topics we'll be discussing today. First, we'll go over the modern cell theory and exceptions to that theory, types of cells, the prokaryotic cell structure in detail, and we'll go into a brief introduction to the eukaryotic cell structure, and then we'll look at a slide that shows the key differences between prokaryotes and eukaryotes. Okay, so the modern cell theory states that all living things are composed of one or more cells. The cells are the basic structural units of life, and they're made from pre-existing cells. The cells are the same in terms of the chemical composition, and the hereditary information is passed on from one cell to another through division. There's also energy flow within the cells, and this occurs through metabolism and biochemistry. Okay. The two big exceptions to the cell theory are viruses. These do contain cells, however, they are not alive because they are not able to grow or reproduce outside of a host cell. Also, spontaneous generation is another exception. It was once thought that cells did in fact arise spontaneously, but however we know that is through cell division. Okay, the main two types of cells we will discuss today are prokaryotes, and they're also known as bacteria, and eukaryotes, which are animal and plant cells. Okay, for a prokaryotic overview, they are the simplest organisms. They contain a rigid cell wall and have a cytoplasm surrounded by a plasma membrane. They also have rotating flagella. This is found in some bacteria, not all. Okay, this next slide shows you a picture of a common structure bacterial cell. So, taking a look at the prokaryotic structure, first we will look at the cell wall. The cell wall in bacteria is made of peptidoglycan, and this consists of carbohydrate matrix, which is cross-linked by short peptide units. So, looking at this picture here, we can see that the cell wall can be classified as either gram-positive or as gram-negative, and this depends on how the bacteria is going to react with certain bacterial antibiotics. So gram-positive is generally thicker cell wall, thicker peptidoglycan, whereas the gram-negative bacteria are very complex and they have less peptidoglycan. It's a bit thinner. So the cell wall, it not only protects the cell, but it also keeps from excess water entering the cell and bursting, and it also keeps a shape. So this next slide, we're going to take a look at the cell wall gram stain procedure. So we have gram-positive and gram-negative. The first step in this procedure is to fixate the cells on the slide. Then we add the crystal violet, and this stains both cells purple. The iodine treatment fixates the crystal violet onto the cells. Then we have a discolorization. So in gram-negative, you'll notice that the crystal violet is washed away off the bacteria, whereas in the gram-positive, the crystal violet stays in the cell. So the next step is the counterstain, and that is safranin, which is a reddish color. The gram-negative takes on the safranin and stains red, and the gram-positive still keeps the crystal violet unaffected by the counterstain. So gram-positive under a microscope looks purplish, gram-negative looks reddish. Next, we will take a look at the cell interior of the prokaryote. So, as we said before, bacteria is a very simple structure with a plasma membrane. But within the cytoplasm, which is the oily type substance in the cell, it contains ribosomes as well as a bacterial chromosome. So the membrane, it acts similar to the membrane-bound organelles that are found in eukaryotes, but we will discuss that later in an upcoming lecture. The plasma membrane also facilitates cell division, And then the ribosomes inside the cytoplasm also help facilitate protein synthesis. And in terms of the hereditary information, the chromosome is a bacterial, simple-shaped DNA. And of course, as we said before, there are no complex membrane-bound organelles found in prokaryotes. And the next main structure of prokaryotes are flagella and pili. So flagella act sort of like a motor to help facilitate movement in the bacteria. They're made of protein fibers, and prokaryotes may have one flagella, they may have several, or they may have none. There's also pili that may be found in prokaryotes, and these help transfer genetic material from one cell to another. Okay, this next slide, we're going to briefly take a look at eukaryotes. So as we said before, they are a lot more complex than prokaryotes. They are included in animal cells and plant cells. The basic structure consists of a plasma membrane, a cytoplasm, a cell wall, which is only found in the plant cells, not typically in animals, the cytoskeleton, and the organelles. So this slide, as you can see, the main organelles and some of the differences in how the animal cell looks versus the plant cell. So this is just to give you an idea of what's to come in an upcoming lecture. This final slide we have here is going to give you the differences between prokaryotes and eukaryotes. Sometimes it's a good idea to look at differences side by side. As you can see, cell wall is found in prokaryotes. However, it's a protein polysaccharide. It's also found in eukaryotes, but in this case, in plant cells, and this is composed in cellulose. So those are two main differences. They both have cell membranes. Both prokaryotes and eukaryotes have ribosomes. However, prokaryotes do not have membrane-bound organelles, whereas eukaryotes do. And in terms of hereditary information, the prokaryotes contain one circular DNA, whereas eukaryotes may contain many. All right, that's the end of the lecture. Thank you for visiting myotherprofessor.com. And please refer to our website for more lectures. And also feel free to leave some feedback if you have any suggestions or other information you'd like to see covered. Thank you.